Welcome to the Cell and Gene meeting on the MISA, where I'm going to present the great progress we are making at Atara Biotherapeutics for the benefit of patients. After the usual forward-looking statement, I would like to highlight that the key mission of Atara is to transform the lives of patients with severe disease through pioneering science, teamwork, and a commitment to excellence. We're doing so through a very unique and differentiated platform, which is the most advanced allogeneic T-cell platform right now, with demonstrated feasibility of delivering treatment to patients within 3 days, with a robust, scalable, and manufacturing process. And we've done so with over 300 patients already treated in our various clinical studies. We are developing this platform for different types of assets, but they're all leveraging the unique properties of Epstein-Barr virus T cells or EBV T cells from healthy donors. And these EBV T cells are really offering significant advantages in terms of safety, as they are very specific to EBV antigens. They don't have much alloreactivity. And also in terms of expansion, trafficking, cytotoxicity, and persistence. Very unique and differentiated type of cells that we're leveraging in our various platforms with two types of assets. Some of them are really addressing EBV-driven disease. And that's the case of Tab-cel or Tab-cel in EBV-associated cancer, and also of ATA188, or unique and very specific EBV T-cell immunotherapy for multiple sclerosis. Here, in that case, we're really leveraging the endogenous TCR of these cells. But we could also insert in this allogeneic EBV T cell a chimeric antigen receptors, and we have done so already in developing two assets and many others coming, one called ATA3219, which is an allogeneic CD19 CAR T, where we have inserted the CD19 CAR with next-gen technologies in terms of co-stimulatory domain into this allogeneic EBV T cell, or also ATA3271, or allogeneic CAR T targeting mesothelin, and hence to be developed in solid tumors. Both of these assets being in IND-enabling studies as we speak. All of this is being made in our dedicated state-of-the-art manufacturing facility in Thousand Oaks, California, which has already been qualified for clinical trials. And we are right now making commercial batches for the validation of the testing of these batches for the forthcoming initiation of a BLA for Tab-cel. We are leveraging all of this platform with three key priorities: Tab-cel, Tab-cel, ATA188 in MS, and our CAR T program, allogeneic CAR T program. Tab-cel is addressing first a unique indication, which is relapsed refractory PTLD, which is a very severe and deadly cancer arriving in patients post-transplant. And in the case these patients are relapsed or refractory after first-line therapy, the median survival is just a few months. So here we have already data from our phase 2 program with Memorial Sloan Kettering, as well as our expanded access program that shows that we are able to obtain not only a high level of response in these patients, but also the duration of response. And this then leads to very significant overall survival at 2 years in responders, over 80%, be they patients that are coming from stem cell transplant or from solid organ transplant. So very encouraging early result with Tab-cel in these patients. Hence, we believe that we have the potential to create a significant and compelling value proposition for these patients as well as for the healthcare system. Not only having high and durable treatment effect, but also an excellent safety profile. And at this stage, we have no CRS experience in these studies and no PTLD-related mortality. This is coming with a low cost of administration. No pretreatment is required for Tab-cel, 5 to 10 minutes IV infusion, and just a 2 hours monitoring. And of course, as I said earlier on, these cells are truly allogenic off the shelf and can be delivered to patients within 3 days. Now, this is important because this allows us to address an ultra-rare disease, but with a compelling value proposal. And this is really trying to address the need of several hundred EBV positive PTLD patients every year in the US who have failed first-line therapy. Now, we don't stop here, and we're just starting a new study in different type of potential new indication for Tab-cel to expand its potential. And this is our multi-cohort study that is addressing up to 6 additional potential indications for Tab-cel. And we are going to have the first patient enroll in that study by the end of the year. This study is particularly important in 2 cohorts for which there is a significant number of patients, but also recently we presented some early data from our expanded access program in this population of acquired immunodeficiency or primary immunodeficiency lymphoproliferative disorders. In these data presented at ESMO very recently, we've been able to show that in these patients, we have not only a significant and a clinically relevant response rate, but we also see some very encouraging data from an overall survival point of view. And all that with an acceptable tolerability and safety findings that are consistent with previously published data. Very encouraging data, and that's also important due to the size of that population. Here we have a few thousand potentially addressable patients with this first-line or second-line situation of lymphoproliferative disorders coming from immunodeficiency, acquired immunodeficiency, or primary immunodeficiency. So that's for Tab-cel. Now moving on to AT188, or EBV T-cell immunotherapies targeting multiple sclerosis. The reason we are using that in multiple sclerosis is because there have been a lot of evidence coming over the last few years on the link between Epstein-Barr virus infection and multiple sclerosis. And that means that having the possibility to eliminate, especially in the central nervous system, the infected cells with EBV that may be, or the trigger of the progression of symptoms, might indeed not only impact positively on the patient, but also could be a way to develop what I would call a truly transformative therapy in MS. What do we mean by that? We mean that today in progressive MS, the current therapies that have been approved are just delaying the progression. What we have here is the possibility to go beyond that and develop a truly transformative therapy that could halt the progression of disability or even reverse in some patients the disability progression. And that will be a real transformative therapy. We believe that AT188 is a therapy that is still in clinical trial, but has the potential to transform MS therapy due to its very unique, very precise targeting of EBV antigens carried by cells infected by EBV in MS patients, and having the potential in that case as an off-the-shelf, easy-to-use type of cellular therapy for MS to improve the disease in progressive MS. We've recently presented an update on a clinical study we are conducting called the phase 1A study for AT188, which is not only looking at safety with 4 different dose cohorts, but also some early sign of efficacy, and through an open label extension to look at what's happening in the long term in terms of both safety and efficacy. At ACTRIMS very recently, we presented an updated data set, especially looking now at all the 4 cohorts in terms of a very important parameter of clinical efficacy called sustained disability improvement, which is the ability for the therapy in this patient to not only show improvement in disability, but showing that in 2 consecutive time points, so having a truly sustained disability improvement in this patient. And you can see on this slide that in higher dose cohort 3 and cohort 4, we had a significant number of patients out of 6 patients per cohort where we demonstrated some sustained disability improvement. So very encouraging results from the point of view of having a therapy that potentially could reverse the progressivity of symptoms in these patients there. Now, the conclusion of this study as presented at ACTRIMS were very clear that not only we have some early clinical sign of efficacy, but this is also clear that the highest dose cohort have a more impact on SDI, on sustained disability improvement. And we also presented the early results from the open label extension. And this is showing that every time we had a patient that obtained the status of sustained disability improvement with the therapy, this sustained disability improvement was maintained at all time points, and even beyond 1 year at 15, and even for one patient at 18 months. So these are very encouraging data of the potential for this therapy to change radically the way MS is being treated today. Of course, this needs to be confirmed in a randomized controlled trial, and we are starting that randomized controlled trial right now. We've enrolled the first patient, and we've recently decided to increase our investment and update the endpoint to take sustained disability improvement as the primary endpoint of that study, where we compared placebo to AT188. And here we will also consider making some updates on the protocol with the highest dose cohort, as well as maybe extend the follow-up of this patient. But this study is extremely exciting, and we believe it will be able to demonstrate, hopefully, if positive, the potential of AT188 to truly transform the treatment of MS in the years to come. If we are successful here, this is a significant potential for this company, in the sense that transformative therapy in progressive MS is truly a potential multi-billion-dollar opportunity. If we look at not only the current market size right now, but the fact that having a truly transformative therapy that could alter or reverse disability will lead to a higher treatment rate, and of course, having a significant market share, this means that such a therapy could have a potential annual US revenue opportunity just in progressive MS or over 3.5 billion. So very exciting time indeed for Atara and our program in MS. Moving now to CAR-T, we had recently cleared the IND for ATA2271, which is a dedicated new next generation CAR-T that is targeting mesothelin. And as you know, mesothelin is overexpressed in a number of solid tumors, such as not only mesothelioma, but also non-small cell lung cancer, ovarian cancer, and pancreatic cancer. And here we have not only a CAR that is targeting mesothelin, but a CAR that is benefiting from very new type of technologies, including a new costimulatory domain called 1XX, and also a PD-1 dominant negative receptors that is designed to provide intrinsic checkpoint inhibition. Recently, we presented preclinical data at AACR 2020, clearly showing that combining these new technologies allows not only to maintain a good safety, but in animal models to improve the functional characteristic and to have these cells being able to functionally have the possibility to expand and eradicate tumors after several, up to 10 tumor rechallenge. So very exciting time indeed for that program that is now entering the clinic. We've also another program in IND-enabling studies that hopefully will enter the clinic next year, and this is ATA3219. This is our first allogeneic EBV CAR-T targeting, in that case, CD19. And here we use the natural biology of this EBV T cell. We insert a CAR with these new next-gen technologies, especially the novel 1XX costimulatory domain. And the idea here is to develop an allogeneic CAR-T that could be the best in class in that category. We also benefit from previous data shown recently in the Congress that have evidence that this type of allogeneic EBV CD19 CAR-T could lead to a high level of response and a very long duration of response over 2 years. So we're building on that with our program, Atara program called ATA3219. Now, if you look at all our program, the Tab-cel, AT188, ATA2271 in addressing mesothelin, and the allogenic version, ATA3271, as well as our CD19 CAR-T, allogenic CAR-T, ATA3219, they all benefit from this natural biology of EBV T cell, and therefore from the platform that we have built from a process science, a research, and manufacturing point of view. So we are now delivering on expectations, and over the next few months, we hope to make sure that not only we begin the enrollment in a phase 2 multi-cohort study, including patients, as I said earlier on, with other EBV positive cancer, but also engage with the FDA with an interim analysis of our pivotal study for Tab-cel in relapsed refractory PTLD, and following, pending a pre-BLA meeting, having the possibility to start initiating a BLA submission by the end of the year. So here we are with all of this progress made at Atara Biotherapeutics for patients and for our company. Thank you very much for your attention.